All right. Hello, everyone. We're going to do a little screencast on chapter 11, which is genetics. This is going to be the first day of information. I'm going to try to section this off into the different days that I go over just the material in general, just to make it a little bit easier on you to see which content or which PowerPoint slides are going to be in each day if you're not in class. So again, this is day one. All right, genetics. This is just the whole branch of science. It's going to look on how characteristics are passed from one generation to the next. You can see we're going to be going over genetics in this whole chapter. So it's a pretty beefy chapter. There's a lot of material in this. But yeah, this is just the whole field of genetics. A specific gene is going to be an inherited factor passed on from parent to offspring, and these are going to be located in the DNA of your chromosome. So we have this chromosome here. You can see the chromosome is made of DNA. The DNA, a section of this DNA, is known as a gene. So these genes are going to specifically code for proteins that are going to make us who we are. And we're going to be talking a little bit more about that in the next chapter. So again, this is kind of like it pushes forward here. We're going from meiosis. Now we're going to go from into genetics, and then we're going to go into how these genes then produce our characteristics or our proteins. All right, so these genes have different alleles, and these are just different versions of the genes, and they're represented by a single letter usually. In this class, they definitely will be represented by a single letter. You can see we have different genes for like eye color. So you have like the blue eye gene, the brown eye gene. We have different hair color genes, like if you have dark hair, light hair, curly hair, whatever. We have different alleles for these genes. So different versions of the same gene. They're still the same gene in the sense of like, yes, you have like eye color genes, but they have different alleles like blue eyes and brown eyes. All right, these alleles lead us to our genotypes, and the genotypes are just the set of alleles or the combination of alleles that you inherit from your parents. Remember, you inherit one of these chromosomes from dad, one of these chromosomes from mom, and these pairs of chromosomes are known as homologous chromosomes, and these pairs of homologous chromosomes will have these allele combinations known as genotypes. The example of a genotype are two letters like big B, big B, big B, little b, or little b, little b. Again, you can see these alleles are represented by, or I should say, these alleles on the homologous chromosomes are represented by these two-letter combinations. Now, these alleles are going to code for your phenotype. So the combination of the alleles, the genotype, will code for the phenotype. The phenotype is the physical characteristic. So again, you have your chromosomes that have DNA with the genes on them. These genes are going to code for what you look like. And not only what you look like, everything inside you, everything, even like your brain structure. Now, there will be a lot of environmental factors that play into who you are and how you act, but they also do code, like these genes do also code for the ways that you act as well. So again, phenotype is your physical characteristic. Now, we have two combinations of these allele or genotypes. We have homozygous and heterozygous. Homozygous means they have identical alleles. The alleles are the same. Another name for homozygous is purebred. So you can see big B, little b, little b. Just a little kind of note here. We don't like abbreviate this. We have to say the whole word. Homozygous. Heterozygous, two different alleles. These are when they have different. So again, homozygous means the same. You can see big B, little b, little b. They are the same. Heterozygous, that means they're going to be different. Big B, little b. The capital letter always comes first in this case. These are also known as hybrids. So heterozygous, two different. Homozygous, two identical. Just a pair of heterozygotes. Oh, come on, that's allele. Get it? A little allele funny. All right, these genotypes and the alleles will either be dominant or recessive. Dominant, as you may kind of get from the name, they're the stronger of the two genes expressed. And what's going to happen is we represent these by capital letters. The recessive genes or the recessive alleles, these are genes that are going to be masked by the dominant allele, and you can guess again, lowercase letter represents the recessive allele. So again, dominant, they're the stronger. That's what's usually shown. Recessive, that's the weaker of the genes. These are the lowercase alleles. So big B. Is this a homozygous or is this a heterozygous genotype? This is a homozygous genotype, and the big B's mean that it's a homozygous dominant genotype. We have to say whether it's a homozygous dominant or homozygous recessive, because again, these are homozygous. They're both homozygous, but this one's recessive and this one's dominant. This one right here, big B, little b, is heterozygous. Now, we don't have to technically say whether it's dominant or recessive because we know it has to have one of each. It's just going to be heterozygous. Now remember, your genotypes are going to code for your phenotypes. So again, genotypes code for phenotypes. Now, for Mendelian genetics, this is just for Mendelian genetics. When the dominant allele is present, the dominant phenotype will be produced. I'm going to say it again. The dominant allele, when it's present in Mendelian genetics, the dominant phenotype will be produced. So again, we have this big R, purple flower, little r, white flower. Whenever we have a genotype of big R, it's going to be a purple flower. It has to be a purple flower. Big R, little r, it's going to be, again, purple flower because we have at least one of these dominant alleles. Now, you might say, Mr. Fradoff, what about little r, big R? It doesn't matter what this combination is. If it's heterozygous, you put the capital letter first every single time. Again, when it has one of the dominant alleles, it's going to have the dominant phenotype. And then when we have the little r, that's just going to be the white flower because it doesn't have any of the dominant alleles. So again, little r is going to be white. Big R, little r is going to be purple. So this is Mendelian genetics, and this is going to be the end of day 1, actually. So if you're at home and you're doing the notes, this is the end of day 1. This is probably my favorite meme of all time. This job isn't for everyone, but hey, it's in my genes. Gotta love the scarecrow here. So what I would do is stop here for day 1, and then we'll talk about day 2 next. All right, so this is going to be day 2 of information on genetics. It's not a big day because there's not a lot of notes, but it's really important that we understand Punnett squares going forward into the next day. So Punnett squares, what do they do for us in genetics? Remember back in the last chapter, we talked about meiosis and how meiosis creates sex cells with only one chromosome per pair of chromosomes. So we have this homologous pair of chromosomes. You can see there's 2 here. This chromosome might be from your mom. This chromosome might be from your dad. Now, you're not going to pass on both to your offspring. We only pass on one of these to our offspring. That's because meiosis produces sex cells with only one chromosome per homologous pair, which means we're not going to pass on both of these alleles because remember, the alleles come from your parents. These alleles are in your homologous chromosomes. You don't pass on both alleles. You only pass on one. So each letter is going to represent an allele that might be passed on to your offspring. So again, you receive these 2 chromosomes. You're only going to pass on one. So what we have to do is we have to make these things called Punnett squares. And these Punnett squares help us predict the chance of an offspring having certain traits. Remember, letters represent these alleles in these Punnett squares. So let's get into it. This is a Punnett square. You may have done one before. I'm going to quickly kind of go over how to get through one of these. But basically what you'd have to do is you have to show that this Punnett square, you have the parents on the top and you have, let's say, like one parent here, one parent here. And all you got to do is you got to bring down each letter and fill it in each box. So this little a goes here and here. This little a goes here and here. And it looks something like this when you're done. Now, big A goes here, big A goes here. That means this offspring or this potential offspring is homozygous dominant. Even though the parents are big A, little a, they have a 25% chance of having big A, big A. That's because out of these 4 boxes, each one of these represents like a zygote or a possible zygote. There is a 1 in 4 chance of being homozygous dominant. This box is heterozygous. Remember, we always put that big A first, even though you have a little a here, we put the big A first. There is a 2 in 4 chance of being heterozygous. You might be like, why is there a 2 in 4 chance? There's another heterozygous one down here. So we can get this combination or we can get this combination. They're the same genotype, but they're just from different alleles. So you can see this A is here, this A is here. They're the same genotype though. So again, 2 out of 4 is 50%. The offspring between these 2 parents have a 50% chance of being heterozygous. And the last one here, you have a 25% chance of being homozygous recessive. So what you're going to do is you're going to practice with making these Punnett squares. There's going to be, I think it's your second page in your packet. You're going to be working with Punnett squares the whole day with just this simple moving the alleles around. So this is the end of day 2, actually. All right, so the third day here in the Mendelian section, and I actually think I'm going to divide this chapter up a little bit in with at least of the video to make it a little bit more reasonable because I think I'm already at like 12 minutes for maybe 10 minutes for the this video. This is day 3 of Mendelian genetics. We're going to go over Mendel. So we just got through with like alleles, genotypes, and phenotypes, dominant recessive, homozygous heterozygous. I was on day 1. Second day, we did work with Punnett squares. This third day, we're going to look at Mendel's explanation of genetics and what he found out through his data. So today is going to be the last day of Mendelian. It's called Mendelian genetics. We're going to then move on later to something called non-Mendelian genetics. So in Mendelian genetics, we talk about before Mendel came around. This is pre-1800s. People thought that the blending explanation, that blending hypothesis, was how traits were passed on. And what the blending hypothesis states is that the traits of two individuals will blend together in their offspring. So, like if you had a really tall person and a really short person have kids, they're going to be median height. And there is some evidence that supports this. And yes, sometimes when you have a shorter person, a taller person, their kids are like medium height. So you think, okay, everything blends, but that's just not how everything works. Another example is like a red flower mating with a white flower, they get all pink. If you think about it though, two pink flowers would make more pink flowers, and then there wouldn't be any red or white eventually because all the flowers would somehow be pink after many generations. So we understand this just isn't correct. And Mendel disproved this hypothesis. He knew something else was going on. You can see when he lived, he was an Austrian monk, which means he was a religious person who worked, he studied and was at I forget the actual town name, but he was working in, if I'm correct, Czechoslovakia, I think was it Austria? I forget. It's one of those countries out there. But he was educated in science. He studied physics and botany at the University of Vienna, and he is known today as the father of genetics. Okay, so Gregor Mendel. So he took 8 years. He knew that something was being passed on from parent to offspring. He just couldn't tell what it was or how the patterns of this inheritance were passed on. So what he did was he worked for 8 years. He worked with thousands of pea plants. I mean, thousands of points of data. This is really a time when science was just kind of its baby years. It was just developing the scientific method and using math and how to use math. And he found really cool patterns in these peas. I always think that Mendel looks like Dwight from The Office, but I don't know. Maybe it's just me. This is his garden. This is in I forget the country now. I want to say it's Czechoslovakia, but I can't. Yeah, it is Czech Republic. Okay, good. So yeah, so this is where he did his research at. You can see here his garden and what it looks like today. There's a big statue there, but he looked at different traits of pea plants. And the great thing about pea plants is they reproduce pretty quickly. Not like humans that take like years and years to reproduce. And a lot of the phenotypes in plants are very easily identifiable, which means you can say yes or no, they're this or that. So like yellow versus green pea pods, inflated versus pinched pea pods. And he saw these traits were being passed on from parent to offspring. So a couple our terminology words here that we have to know. P stands for the parental generation. That's like if the P generation for you would be like your parents' generation, that would be like that's P generation. You are the F1. That's the first generation below the parent generation. So like you and your brothers or sister would be the F1. The F2 would be the generation below that. So again, that'd be like your offspring, your kids that you would have someday. So again, P, F1, F2. If your grandparents are the P generation, then your parents would be the F1 and you would be the F2, and then your kids would be the F3 and so on and so forth. It all is determined by where you put that P generation at, though. Now, what Mendel did was he kept on crossing certain types of flowers or plants or pea plants, whatever, with themselves. So like purple, we're going to use an example here, purple flowers. So he kept on crossing true breeding. So he just kept on crossing these, and he also kept on crossing like the white flower plants. This was known as the pea generation. What he did then was he crossed a purple and a white flower. And he thought, well, if the blending hypothesis is correct, you should get light purple. And that's not what he found. What he found was all the offspring were purple. And that's the F1 generation. So let me show you what that looks like. So he had a true breeding that was just producing a bunch of purples and a true breeding white that was producing only whites. And he crossed them and he only got purple flowers. And that didn't make sense to him. He's like, these two should make light purple. So what he did was, in the F2 generation, he crossed two of these purple flowers. Now, yes, it's a little bit of inbreeding in plants, but plants work a little bit differently. And what he found was that the F2 generation had a 3-to-1 ratio of purple to white. Let me show you what that looks like. So again, we cross these two initially. They produce purple flowers. Then we cross two of these together, and they get a 3-to-1 ratio of purple to white. And he's like that, and he kept on getting it. It's not just for purple versus white flowers. He got this in every sort of, I think the next slide's data. Yeah, he got this in every, like when he crossed purple flowers, axial versus terminal, yellow, every one of these characteristics, he kept on getting that 3-to-1 ratio. Now, he knew something was going on. He knew that something was being passed on from generation to generation, and he published his work in a natural history journal in 1866, but it was just like kind of like a dud. Like it was like, okay, it's something's going on with these pea plants. His last words were, "My time will come." He knew how significant his data was, and his results actually were forgotten until the 20th century. And when geneticists in the 20th century were like, "Oh, like I remember reading something about this in some pea plant some guy did in like the 1860s," they realized how beyond his time he was. So like he understood genetics are being passed on from generation to generation far beyond 20, 40, 30, 60 years before people were really understanding this. So what we're going to do in class, and I love this meme too, what we're going to do in class is we'll have you look at some of Mendel's data and see if you can come up to the same conclusions as he did.